But this leads us to a much more general question. So what is the best model for the hemodynamic response function? If I were to present a participant with a stimulation, right here, how do I expect the brain to respond? Which function best approximates what somebody's brain might do in response to this? So if this were my stimulus, right, what we've been doing is what people often refer to as the canonical HRF. We've been saying, look, if you present a participant with stimulation, let's just say, a picture of a face, then we expect the brain to respond sort of in this fashion, gamma. Or maybe in this fashion, a double gamma. And see, the key intuition behind the canonical hemodynamic response function is that whichever exact function, whichever exact sort of shape you pick, the idea behind it is that most of the aspects of this shape are fixed. For example, the initial dip, the time it takes from baseline to initial dip is fixed. The time it then takes from the peak of the dip to the maximum of the function, that is fixed. The time it takes from the peak to go back down to the bottom of the undershoot, that is fixed. The time it then takes from the bottom of the undershoot to go back to baseline, that is fixed. In other words, the shape of the canonical HRF, be it a double gamma, as I'm showing here, or a simple gamma, that is fixed. Everything is fixed. The only free parameter is the extent of the response, is the amplitude of the response. Now, this is, on the one hand, it's wonderful because it reduces everything that we're doing to one parameter. What is the size, what is the amplitude of the response? But on the other hand, as we're going to discuss in just a moment, this approach is highly biased because this approach works well for all the people whose brain responds exactly in this way. If your brain happens to have, let's say, a shorter interval to peak, then you'll be penalized because your hemodynamic response function won't match this fixed shape. So again, this is wonderful because it gives us one parameter, and that parameter is easy to interpret. It's even easy to aggregate at the second level when I'm bringing together analysis from different participants. I have one parameter that I put together and I can average across participants to get a group value and a group test. On the other hand, I am highly biased. If somebody has a big response to a stimulation, but the shape of their brain, the shape of their hemodynamic response function is not exactly the one that I'm expecting, or the one that I'm positing, then they will be penalized and it will look like they have a lower response as compared to somebody who has the exact same amplitude, but their HRF also matches perfectly the shape that I'm using, in this case, a double gamma. So in the case of using a canonical HRF, our GLM would look something like this. And again, I'm simplifying in the sense I'm excluding from this design regressors of non-interest. So we have our time series, and what we expect the brain to do is something of this sort. And we say, well, there's a trial here. In fact, there's a block here. So we expect the brain to kind of, from baseline, kind of shoot up, then come down and down again, then be down during the baseline task, and go on and off, etc. And of course, as I was highlighting earlier, we have only one parameter, which makes it very practical, as I said, for second level analysis. Now, is the canonical HRF a good approximation of what people of what are the actual hemodynamic responses in participants? Not exactly. In fact, there's a lot of published data to show that individuals vary significantly in their hemodynamic response function, or rather in the shape of their hemodynamic response function. So if we look at the leftmost image, you can see here, see the dashed black line? That is the canonical hemodynamic response function. All the other lines are participants under varying sampling conditions, but are participants. Now, there certainly are some participants who have whose response function, for example, the green one, approximates pretty well the canonical response function. But there are other participants, for example, this purple one, who has the exact same amplitude of the response. See the deflection from baseline to response has the same amplitude, but very different shape, right? It has a much longer lag at the beginning. So even though it has a big amplitude, just because it's different than the shape we expect to see, the participants will be penalized. It will look as if they have a lower response, just because their shape doesn't fit nicely the canonical HRF. And this is Handwerker 2000 and colleagues 2004. In 1998, Aguirre and colleagues did a very similar study. And you can see, again, there's a huge variability across individuals in their HRF response as compared to the canonical HRF. Now, not only there's a lot of variability across participants, there's even a lot of variabilities within participants. In the sense, if you take the same participant and you have them do the same task, just in different runs, and this is the left graph, you can already see some differences. These different colors represent different runs. So there already are some differences for the same participant, same task, different runs. And on the right, you're seeing the same participant doing the same task on different days. And you can see that now there are, for example, this purple and the green lines and the cyan lines clearly deviate from the other ones. So not only there's a lot of variability across individuals, there actually is also a lot of variability within individuals, both within a single day and across multiple days. And in fact, it gets even worse. Even within the same individual, you can actually get different parts of the brain that look very different. So each of these little graphs is a different subject. And the different colored lines, so we still get the canonical in the black, in the dash dot dash line. But then the other lines are just just different parts of the brain. For example, we have the frontal eye fields, motor cortex, the supplementary eye fields, and visual cortex. So we have prefrontal, frontal, and occipital cortex. And for example, if you look at perhaps the most dramatic is subject 11 at the bottom right of this graph, you can see again the canonical shape right here. And then look at how different V1 is with this giant dip down here. And even the other ones are still very different. For example, this is the frontal eye fields, supplementary eye fields in red, and then M1 in green. So even within the same subject, not only there's a lot of variability within and across days, but even across different parts of the brain, there's a lot of variability. So the canonical has a lot of has some pros and some cons. And to put this together, on the plus side, using a canonical HRF, just like a double gamma, has some significant advantages. It gives you one parameter. It's very easy to interpret. It's the amplitude of response to stimulation. And it's very easy to integrate and to aggregate at the second or third level, meaning it's easy to take for every one of my participants or every one of the runs of every one of my participants, one parameter, beta, and bring it all together in one large group analysis. It's very simple to do, it's easy to interpret. On top of it, because we're imposing a certain shape, because we're biased towards a certain shape, it typically results in less variance. However, we are biased to find activations that have the specific shape of our canonical HRF. If somebody, as I was saying earlier, happens to have a big amplitude of response, but in a different shape, it will look as if they don't have a strong response. So we're biasing ourselves towards finding responses that have the shape of our HRF. And anybody who doesn't match that will get penalized in terms of the resulting amplitude, or rather the resulting magnitude of beta. And finally, given all the data I've shown you about variability within the HRF, it's highly likely that if we just use a canonical HRF, we are mismodeling what's happening. So a different way of doing this is, instead of just using one canonical HRF, is to use what are known as basis functions, which means instead of using one function, we actually use multiple functions. In a sense, the canonical is an extreme case of a basis function. It's a set of basis functions that has only one function. Now, the more typical approach is to use two. And this is, if you've done any FSL analysis, typically for eventuated designs, you have used a basis function that has a canonical and its first derivative, also known as the temporal derivative. And this models slight changes in the time it takes in the sort of rising part of the function. So in other words, instead of just using one function, this blue function right here, we use two separate functions which together should better approximate or are flexible and can flexibly approximate somebody's individual response. So as I said, we use the canonical plus we use the first derivative, or the temporal dispersion. The result is that, for example, if somebody has a slightly skimpier or faster rise in their HRF, then there will simply be a greater value in the first derivative, and that plus the canonical HRF will result in that one participant's specific HRF. So in other words, what we're doing is, instead of saying your response has to look like the blue line, we're saying, well, we can shift the blue line to better match your specific HRF by using a combination of two functions. So instead of using one function to approximate your response, we use a combination of two functions. And this is wonderful because it gives us much more flexibility. It allows us to move around the blue function. For example, in this case, they're using a little bit of the first derivative plus the amplitude of the canonical, I can create this shape right here. And of course, if somebody had the opposite, right, I could give the opposite value to the first derivative, and now the green one would have a slower peak and would look more like this. Right? So I use two functions together to better model, to allow for flexibility in modeling the HRF.